everyone. I welcome you all to the afternoon session of Emerging Phytonutraceuticals Against Cancer. So chairing the session, I am pleased to invite Professor Gopal C. Kundu. He is the Director R&D at Kalinga Institute of Industrial Technology. His area of interest is tumor biology, cancer stem cells, and tumor-stroma interaction, among many others. Professor Kundu has done his PhD in chemistry from Bose Institute, Kolkata. He has several postdoctoral fellowships awarded to him from the United States. He was the Dean and Director at NCCS Pune, India. He was the Professor of Medicine and Biochemistry at School of Biotechnology. He has over 116 scientific publications, 1 US patent and 4 Indian patents. He has received a fellowship award for research excellence from NIH, USA, and he is the recipient of the prestigious Shanti Swarup Bhatnagar Award from CSIR. The other chairperson, Professor Ajay Kumar, will be joining us in a little while. I would like to request Professor Kundu to please start the session. Okay.
And our area of interest, bioprospecting of natural products having chemotherapeutics, chemoprotective and chemosensitizing potential. And Dr. Ruby will be talking on pharmacological targeting of MAKE-ARK by detecting attenuous melanoma invasion and metastasis through suppression of MITFL. Dr. Ruby also received multiple awards, including National Level Women Bioscientist Award, Fellow of National Academy of Science, Fellow of Academy of Science, Trivandrum, and Global Health Award, and published a large number of papers in peer-reviewed international journal. Dr. Ruby, please.
Thank you for the nice introduction. Let me first thank the organizers, especially Alok and Anju, for giving me an opportunity to present my work here. And my lab, as Dr. Kundu said, is focusing on identifying anti-cancer molecules. And we have identified some compounds having anti-cancer potential, chemosensitizing efficacy, and as chemopreventives. And today I will present the work which we have done on tryptanthrene, a compound we isolated in our lab. The compound was found to be very effective against skin cancer. So a part of the work I will present here. So, yeah, the compound was isolated from Wrightia tinctoria, and the name of the compound was tryptanthrene. And tryptanthrene has been reported to be very effective against other cancers also, breast cancer, leukemic cells, etc. So we conducted a screening in different cancer cells of different origin, and we found that the compound is most effective against skin cancer. This is non-melanoma skin cancer, this is melanoma skin cancer. So we conducted an evaluation of this compound in both the cancer types. So today I will present the work we have done on melanoma skin cancer. So, yeah, since it was found to be very effective against skin cancer, melanoma, we evaluated its efficacy in different melanoma skin cancers and in normal melanocytes. The compound was safe up to 20 micromolar in normal melanocyte, whether it was cytotoxic to all the melanoma cells. And it induces ROS mediated activation of apoptosis in A375. Actually this A375 cell, it was a melanoma cell, this was found to be most sensitive to this tryptanthrene, so we selected this cell line for our further studies. So it induced ROS mediated apoptosis in A375 cells, and different, apoptotic related molecule, all the molecules were, the P53, BAX, T-bit, all this, and there was a downregulation of BCL2 and BCLXL, etc. So it was the mode of apoptosis, the cell death was apoptosis. So we conducted an animal experiment. The corresponding concentration up to this 12 milligram per kg was corresponding to the 3 micromolar IC50. So we transfected the cells with the Luke and we injected orthotopically to the NOD skid mice, and we could find, we can find a very good tumor reduction. This is the control animals, this is the treatment animals. The fluorescent, the luminescence also was very less. So even though this was not a metastasis model, we could find that this orthotopic tumors were, the metastasis to different organs of the mice, and that metastasis was very less in the case of the triptanoid treated group. So these were the, a comparative evaluation of different organs. And the tissues we checked for the expression status of the, we can see the PCNA, the proliferative nuclear antigen was completely downregulated by tryptandrin, and the VEGFR2, then BRAF. BRAF is a very important molecule in melanoma, and it was significantly downregulated by tryptandrin. So since we found that
it's
not moving.
Slides not moving. Whatever you are talking is not correlating with the slides.
So I think the net is not working, right? It is, the net is not. Here? Yeah. Okay. So it has reached here only, right? Yeah. So the compound is inducing apoptotic mode of cell death, and so the tumor reduction study was conducted using the A37 cells transfected with the LEU. So we can see a significant tumor reduction with the concentration equivalent to the 3 micromolar concentration, that is 12 milligram per kg body weight. So even though this was not a metastasis model, we could find that these cells were, the melanoma cells were metastasized to different organs of the tumor bearing mice, I mean the control mice, and this was very less in the case of triptandrin treated mice. So the different molecules related to melanoma, this The CNA is the proliferative nuclear antigen, which is a common cancer marker, and this BRAF is a very important.
Presentation mode.
Is that okay? It is very slow. Yeah, after clicking here, yeah, that's right. Yeah. BRAF is a very important molecule in melanoma. We can see that it is significantly downregulated by this molecule. So, but when I go to this one, how can I click? If I have to click on the left side, I have to see that, right? Yeah, that's what I was doing. Now it's okay. Okay. Yeah. So this is a metastasis model. We injected the mice through the tail vein. So these are we did 2 metastasis models. This one is using, this is a mouse melanoma cell line B16F10. Here we inject the mice through the tail vein. It is black cyst mice we are doing. So the melanoma cells will go to the lungs of the mice. This is the control, and this is the treated group. Okay. This is the NOD skin mice. Here also we are injecting through tail vein. We can see this is the control, this is the treatment group. So these are the tumor nodules produced in the lungs of the mice. These are the treatment group. Here also we can see some tumors, but it is very less. So triptandrin prevents the melanoma metastasis. I have to do like this because I have only when we click on the slide it will come. Okay. Here. So we found that the compound, in the tissue level we found that there is a BRAF inhibition, and most of the melanoma drugs are focusing on, are targeting BRAF. Okay. So since we found a very good inhibition of BRAF by the molecule, we thought that maybe triptandrin also working through this way. Okay. So, and we checked, we could see that BRAF is completely inhibited by, these are different melanoma cells, and the BRAF is downregulated by the by triptandrin in all these cells, and there was a very good glide score also for BRAF, and it was noted that BRAF is, triptandrin inhibits BRAF, mutated BRAF, not the wild type BRAF. Okay, so we thought that definitely the mechanism of action of triptandrin also may be through inhibition of BRAF. Yeah, but we can see here, here this is a mutated BRAF and this is a wild type BRAF, and this is also mutated BRAF. You can see the difference in the IC50. A375 IC50 is 3 micromolar, while the others are 15 micromolar. So there is something else. Definitely triptandrin is inhibiting BRAF, but there is some other molecule which is regulating the anti-melanoma activity of triptandrin. Okay. So we did a microarray. The transcriptomic analysis revealed, it identified 4 sets of molecules. could see that for signaling pathways, MAP kinase pathway, IKT pathway, Wnt signaling and Wnt signaling. And we found that all these four pathways are converging in this molecule MITFM. Okay. So the MITFM, that is the microphthalmia associated transcription factor, is the master regulator of melanomagenesis. So we got an indication that maybe MITF is the regulator in this tryptandrin induced melanoma activity. So we checked the melanoma. The expression of MITFM, this one. Yes. We can see these are the different melanoma cells. Here we can see the MITF expression status is A375 is basal. Okay. So in all the other cells, the MITF is overexpressed. So maybe that is why it is the IC50 of tryptandrin is 3 micromolar in A375 and very high in all the other melanoma cells. Here also we can see the BRAF status. So here it is mutated BRAF, this is mutated BRAF, this is wild type BRAF. So the cytotoxicity of tryptandrin is not dependent on the BRAF status, even though tryptandrin downregulates BRAF. But here it is highly correlated. Here it is basal, here on all the three it is overexpressed.
He can operate. Now we operate from that cell.
Yeah. It is not the question of operating, it is time. You need time.
Okay.
Yeah. I am doing that, but he is standing here. But when it needs time, that's all. Yeah. So we can see different concept the activity of this. This is B16 after tryptandrin downregulates, SKMBL28 downregulates. It and the IC50 concentration. So in each cells, tryptandrin downregulates completely the MITFM at its corresponding IC50 concentration. Yeah. Then to confirm this, we overexpress MITFM in A375 cells. It was 3 micromolar. When we overexpressed MITFM, you can see it is from 3, it went to 15 micromolar. Okay, the IC50 changes from 3 to 15 micromolar, confirming that the regulator is MITFM. Okay. So yeah, this is the A375 neo cell, this is A375 MITF. Yes. Then we conducted an animal experiment using, in the A375 loop. This is only neo cells. Here it is A375 MITF. Even at 45 milligram per kg, that means it is 4 times the concentration of the IC50 dose, there was no tumor reduction. But at 60, it is corresponding to the 15 micromolar concentration, there was complete tumor reduction. Okay, that's the same animals, we can see the difference. Okay, 12. Here this is the transfected cells, only at 60, that is corresponding to the IC50 dose, there is a. But we found that at this 48 milligram per kg, the tissue, the BRAF is completely downregulated. There was no BRAF at all. But still there was tumor. Okay. Yeah, this is the solid tumor model. We can see the different organs, fully metastasized. By in 60, there was no metastasis. Yeah. This was another experiment. That means instead of A375 meters transfection, this SKMBL28 have the same endogenous expression of MITF. Okay, here also the IC50 is 15. So in this cells also, when we injected at 60 only, we got the tumor reduction. Again confirming the role of midterm. This is the exact in SK. So this is just a comparison. Here we can at IC50 concentration there is no metastasis. Here only here this is the IC50 dose, no metastasis. And this is SKML28. This is IC50 concentration, no metastasis. So here is the comparative evaluation of midterm expression status and BRAF status. We can see that the compound completely downregulate both BRAF and midterm. Okay, and as I told you, here we at 60 only we could show a complete downregulation of midterm, but the data is not put here. At 48 itself, we could find complete downregulation of BRAF. So there was several reports that midterm is responsible for the chemoresistance of vemurafenib and dacarbazine. Both are very important drugs for used for melanoma. And triptandrin also downregulates the expression of BCL2. This is a major downstream target of midterm. Here also we got a complete downregulation of BCL2.
But we have 4 minutes left.
Yeah. So this this is for patients also we could see expression of midterm, and MAP kinase pathway is completely regulating this expression, this midterm levels. So we found that triptandrin is downregulating downregulating BRAF and midterm, but ERK and MEK are increasing both in the nuclear and cytoplasmic levels. And when you to confirm that we inhibited this MAP kinase pathway with the UCLO, so this upregulation was stopped and the midterm level was unaltered, so confirming the role of the ERK pathway. So usually when ERK is upregulated, the phosphatase MKP3 is maintaining the levels. But here we found that triptandrin does not interact with the ERK. So there are reports that MKP3 mediated dephosphorylation of ERK is dependent on the kinase CK2 alpha. So we checked whether triptandrin can inhibit CK2 alpha. Both by in silico studies, by colorimetric studies and by Western blot. We found that it is a better inhibitor of CK2 alpha than the standard inhibitor CPA, and so we found that it is inhibiting this very good inhibitor of CK2 alpha. So the mechanism is it is downregulating the midterm levels. It is inhibiting the two enzymes CK2 alpha and the BRAF. So the compound is a very good candidate to be studied against malignant melanoma. So this is the recently published work on this. A part of this work is published in CMLS, and these are the previous reports. This is the non-melanoma work. I did not present it here. So this is my lab. Thank you.
Thank you, Dr. Ruby. It's a good piece of work. I'll start with one question first and then I'll ask others. So, triptandrin, that compound, when you treated A375 or B16F10 or A375, it is suppressing that mutated BRAF expression.
Yes.
What exactly it does? Have you checked that once that expression going down, the status of pattern of mutation also changing? Is there any DNA repair or any other events happening?
We did not check. Only the expression is completely gone.
But whether that like a V600E, that position still exist or is it changing the pattern of?
We did not see that.
And what happened in case of NRAS?
The mutation, we are getting a downregulation of all the VRAF. No, the NRAS we did not see.
Any other question, anybody? Yeah.
Thank you. It's a very nice, informative presentation. I have not a question, it's a query that you have used a mice that is biopsy, right?
No.
What mice?
NOD skid mice.
This is-
Which one?
This you have generated orthodox
No,
model.
yeah, orthotopic skid mice, NOD skid.
So it's a immunocompromised mice.
Yes.
Okay, thank you.
Yeah, the other one, for metastasis model, we used both. Metastasis model, we used the black cyst mice, that is it is.
That is
it has-
normal. That is normal.
That is normal.
So and it's a human cell you have used.
That is not human. That is mice model.
For
So
B16,
both models we used.
for
B16
B16 evidence, he has used biopsy.
Yeah.
And
No,
for
not biopsy. Black cyst.
Black.
Yeah.
C57 BJ.
That is mouse. B16 is a mouse melanoma. And the other also
Okay.
we used the other.
And
model.
A375, he has used skin mice.
A375 we used.
Any other question, anybody? If not, then thank you very much, Dr. Dugi. It's a good